So let's talk about Human Genome Project. This Human Genome Project is called the Mega Project. So which is none other than the sequencing of the entire genome. So in the human beings, we call it as Human Genome Project. It was started by the US Department of Energy and National Institute of Health from US, and later so it was supported by Wellcome Trust of UK and also from other countries like Japan, China, France, Germany. So approximately we estimated we have 30,000 genes in the human genome. So totally how many chromosomes they got estimated is 24. So 24 chromosomes. How this is 24 is, so we have 22 autosomes as well as we know, and we have two sex chromosomes. One is X and one is Y. And the largest chromosome is of first chromosome. And the smallest chromosome is Y chromosome. So Y chromosome is the smallest chromosome, and the largest gene, dystrophin, so which has 2.4 million base pairs. So if you could convert this whole thing into book, you will be having for one page 1000 letters. Like that you will be having 1000 pages. Like that, how many books is 3300 books. So we have to form the database and you have to analyze the data, and next you have to send to various companies. So for their usage, mainly pharmaceutical companies will be using this data for the production of various forms of drug. And we can also analyze various genetic disorders like we know Down syndrome, like various other diseases like Turner syndrome, Klinefelter syndrome, like that. And from this it will lead to the rise of ethical, legal, social issues, LC. So these are the things we are going to cover from this. And after sequencing this, it is more useful for knowing the alteration of genes. Other than this, they also discovered for rice and Drosophila melanogaster. Sinorhabditis elegans, which is a free-living, non-parasitic, non-pathogenic nematode, Drosophila, and they discovered for bacteria, yeast, Sinorhabditis elegans, and fruit fly, Drosophila melanogaster, rice plant, and also Arabidopsis. So these are also sequenced in Human Genome Project. It is in the preceding sections you have learned that the sequence of bases in DNA that determines the gene information of a given organism. So how the sequencing of the genes entirely takes place, how what is the nitrogenous base that is present, how they are sequenced with only 4 nitrogenous bases. The entire we have a sequence for particular protein formation. So we have sequencing for each and every gene. So in other words, the genetic makeup of an organism or an individual lies in the DNA, or an individual lies. If two individuals will differ, then the DNA sequence also should be different, right? Oh yes, if they are related, their sequence will be quite similar. If the DNA sequence with my brother and sister will be quite similar when we both came from the same parents, right? So we can observe those things also. We can observe the different kinds of genes present in the other organisms, like Sinorhabditis elegans we have seen, Arabidopsis, rice, bacteria, yeast, all these things. So at least in some places they are different. These assumptions led to the quest of finding out complete DNA sequence of human genome. So they found out how what and all the coding, like sequencing of the entire human genome, like A, T, C, G, like this. So with the establishment of genetic engineering techniques, Yes, we have southern blotting technique, we have centrifugation technique, all these things. So, where it was possible to isolate, clone any piece of DNA, so they have to isolate with how they will cut the DNA is with molecular scissors. We also call it as endonucleases. And the availability of simple and fast techniques for determining the DNA sequence. So, molecular engineering was, genetic engineering has brought to identify the genome. So it's very ambitious project, sequencing the human genome. It was launched in the year 1990. This is very, very important. And the human genome was said to be. This is a human genome project, HGP, is also called the mega project because that was a bigger project so far ever taken. You can imagine the magnitude and requirements for the project, yeah. Because if we simply define the aims of the project as follows. So it was said to be, we already know, it is 3.3 into 10 to the power of 9 base pairs will be there for one genome. The meaning of genome is one set of chromosomes, which means N. And the cost of sequencing they required is $3 per base pair. Like how many base pairs we have? 3 billions of base pairs we have. So that's why the estimated cost is 9 billion US dollars. The total estimated cost of the project will be approximately, we are not telling the exact thing. So it was in the beginning. So estimated cost in the beginning, the total, so $3 per base pair was estimated cost in the beginning. But the further estimated cost of the project, total estimated cost of this project is approximately 9 billion US dollars, They obtained the sequencing were to be stored in the type of books. Further, if obtained sequence, so this information, if they got to stored in books, so if each page of the book contain 1,000 letters and each book contain 1,000 pages, like that, how many books we need? 3,300 such books would be required to store the information of DNA sequence from a single human cell. Okay, so 3,300 books we need. The enormous amount of data is generated also necessitate the use of high speed computational devices. So that much data we have to analyze, we have to store, we have to analyze, and we have to evaluate the data. For data storage, we need computer and retrieval and analysis as well. You have to retrieve the data. Means again, if you want to see that data, you have to see retrieving and you have to analyze the data. HGP is closely associated with rapid development of new era of biology called bioinformatics. They developed the bioinformatics in that. Coming to the goals of HGP, Human Genome Project. Important goals of HGP are, so identify all the approximate 20,000 to 25,000 genes in human DNA. This is the goal. Determine the sequence of 3 billion chemical pairs. We already have seen 3 billion chemical pairs that make up the human genome. So store this information in database. And we have to retrieve this information and improve the tools for analysis. So first they have told in terms of genes. Next they told in terms of base pairs. So they identified 20,000 to 25,000 genes they have to identify. And at the same time, they have to determine 3 billion chemical base pairs. Chemical base that make up the human genome. The information has to be stored in database. And the improve the tools for data analysis. Transfer of the related technologies to other sectors such as industries. And addresses the ethical, legal, social issues that may arise from this project. So the Human Genome Project is a 30-year-old project. Means it started in 1990 and it ended in 2003. But one thing you have to remember, for the first chromosome, it took 2006, means 3 more years, because that is the biggest chromosome. So the 13-year project coordinated by, so who coordinated this, who started funding for this project is US Department of Energy and National Institute of Health. During the early years of HGP, the Wellcome Trust became the major partner, Wellcome Trust in UK. So additional contributions also came because the budget is too much, right? They have to pay too much while ongoing the project. So if the funds are insufficient, continuously the funding has to be provided by the other countries also, because they can also make use for the database, retrieving of this data, analyzing of the data for their drug development, diseases, knowing the disease, how to improve the humans, all those things. Japan, China, France, Germany, and others also. So the project was completed in the year 2003. That's how it is 13 years project. So knowledge about the effects of DNA variations among individuals, how the people, if all the people will not look similar, which means each and every person is a variant to the normal. This can lead to the revolutionary change, the new way of diagnosing. So treat and somewhat someday prevent the thousands of disorders. Like if you see, so we can early prevent, if we came to know Down syndrome is present to the offspring or not, then we can decide whether the mother can abort the baby. Like it is the decision from the mother end. But we can find out early, in early way. We can find out the muscular dystrophy, myotonic dystrophy in early way, right? So beside providing clues to understanding the human biology, learning about the non-human organisms also of DNA sequencing can lead to understanding their natural capabilities that can be applied towards the solving challenging in healthcare, agriculture. We can improve the plants if we know their genome, and we can energy production, environmental remediation, how we can save this environment, means by decreasing the pesticides, insecticides, weedicides. So completely we cannot eradicate some of the organisms. Many non-human models are also seen, such as bacteria, yeast, and Caenorhabditis elegans, which is a non-pathogenic nematode, free-living non-pathogenic nematode, and Drosophila, the fruit fly, and we have the rice, and we have Arabidopsis. We have been sequenced. Other than this, these things are also sequenced. So we'll repeat with me once again. Other than this, they sequenced bacteria, they sequenced yeast, right? They sequenced bacteria, yeast, Caenorhabditis elegans, and Drosophila melanogaster. And they also have seen regarding the rice and Arabidopsis. These things are also sequenced. So let's see what are the method they use. They use two methods. One is EST. This means expressed sequence tags. So they involve two major approaches. One approach is they focused on identifying all the genes that are expressed as RNA. So whatever the genes that are expressed as RNA, they call that gene as expressed sequence tags, EST. Like GST, you can remember the EST, express sequence tags. And the other, a blind approach, they have to sequence the whole set of genome, like coding, non-coding, or the later assign different regions in the sequencing with functions. Later they will differentiate what will function, what will not function. For this, they use the term called sequence annotations. So EST is one, expressed sequence tags, and SCA, sequence annotations. For sequencing, the total DNA from the cell is isolated. So how they sequenced is, they isolated the total DNA and converted into random fragments into smaller size. We recall that DNA is a longer polymer. Okay, and there is no technical limitation in sequencing very long DNA. There is no limitation, right? So they cloned into suitable host using specialized vectors. They used two chromosomes, BAC and YAC, bacterial artificial chromosome and yeast artificial chromosome. So the cloning resulting in amplification of each piece of the DNA fragment. So that's why they subsequently could sequence with ease. So the commonly used hosts were DNA, were bacteria and yeast. So the vectors were called VAC, bacterial artificial chromosome, and yeast artificial chromosomes. These are the vectors they used to clone. So the fragments are sequenced using automated DNA sequencers. So automatically the fragments were sequenced by using automated DNA sequencers that worked under a principle developed by Frederick Sanger. So they sequenced the DNA fragment by using the method developed by Frederick Sanger. Right? So remember, the Sanger is also credited for developing the method for determining the amino acid sequence in proteins. So he is the one in biomolecules, you may get this, sequencing the amino acid for proteins. So Sanger is the one who sequenced the DNA and at the same time who sequenced the amino acid of the proteins. So the sequence was arranged based on some overlapping regions present on them. The required generation of overlapping fragments for sequencing. Aligning these sequence was humanly not possible. So they got some sequence that are based on some overlapping regions, again and again how it is coming. So this requirement, alignment is not possible. So that's why specialized computer based program was developed for this. The sequence was subsequently annotated and were assigned to each chromosome, if that sequence is present in all the chromosomes or not. The sequence of first chromosome was completed only on May 2006. You have to remember this, May 2006 only it is finished, because it is the last one of 24 chromosomes, because 22 are autosomes and we have the X and Y, because it took 2006. If you see here, we have to take any sample of hair follicle, blood, we can take the sperms, and we will be extracting the DNA. So after extracting DNA, we will make the DNA into segments. So that segments will be cloned using the vector. Vector is bacterial artificial chromosome and yeast artificial chromosome. And after amplification, so that was easily sequenced by Frederick Sanger method. So another challenging task was assigning the genetic and physical maps of the genome. So first they know the sequencing of the gene, and later they came to know about where the mapping is. That was another challenging task. This is generated by using the information on polymorphism of restricted endonuclease recognition sites. So any one nucleotide will be different in the sequence, that we call it as polymorphism. So the restricted endonucleases would recognize that site and will cut. Some repetitive DNA sequences known as microsatellites. One of the application of polymorphism in repetitive DNA sequence shall be explained in next DNA fingerprinting. They observe these things also, microsatellites. Coming to the next thing, what they observed actually. So what they observed is salient features of Human Genome Project. So Human Genome Project was as follows. They contain 3164.7 million nucleotide bases. So they found out 3164.7 millions of base pairs. The average gene consists of 3000 bases. One gene consists of 3000 bases, but greatly vary in size. And the largest human gene is dystrophin, which consists of 2.4 million bases. Right? The total number of genes that was estimated is 30,000. Almost all 99.9% nucleotide bases are exactly same, because they were previously estimated we have 80,000 to 1,40,000 genes, but we came to know only 30,000 genes are there. But almost all 99.9% of genes are nucleotide base pairs are same, exact for same for all the person. The function is unknown for over 50% of the discovered genes. We don't know what is the function of that. Less than 2% of genome codes for proteins, less than 2% only. The repeated sequence will make up the larger portion of human genome. So only repeated portion you have the larger sequence. Sometimes 100 and 1000 times also. So repeated portions only will make, we call it as variable number tandem repeats. They are thought to have no direct coding functions, but they have shed light on chromosome structure, dynamics and evolution, right? So chromosome 1 has most genes. You have to remember this. Previously we have know one more number, that is how many base pairs is 3164.7 millions of base pairs are there. Here 2968, 2968, and 231 is for Y. So repetitively you have practice, you will remember this number. 2968 is for chromosome 1, and 231, 2968 chromosome 1 and 231 is for chromosome Y chromosome. Because chromosome 1, it took 3 more years to finish, because by 2006 May, because it has 2968 genes. Y has 231 genes, which is smallest, and scientists have identified the about 1.4 million locations where single base pair DNA difference, single base pair only differentiated. They observed at 1.4 million locations. So we call that as single nucleotide polymorphism. We call that also as SNPs. Occurs in humans. Single nucleotide polymorphism. At 1.4 locations they got identified. This information promises to revolutionize the process in finding the chromosomal location, but disease associated sequence. They can find out disease associated sequence and tracing the human history. How we apply the human genome? So let's see with DNA fingerprinting process. So application of future challenges. So deriving the meaningful knowledge from DNA sequence will define the research through coming decades, leading to our understanding of biological systems, means how the systems will work, how those things. The enormous task will require the expertise and creative tens and thousands of scientists from varied disciplines in both public and private sectors worldwide. So one of the greatest impact of having human genome sequence will be enabling the radical new approach in biological research. We can research in many things. In the past, researchers studied only one or few genes at a time, but this whole genome sequence and new high throughput technologies, we can approach questions systematically and much broader scale. We can study all genes in the genome, for example, all the transcripts in particular tissue or organ or tumor. How tens of thousands of genes and proteins work together, interconnect, and to orchestrate the chemistry of life. Next, we have the very interesting thing, that is DNA fingerprinting. We previously have seen SNPs, single nucleotide polymorphism, and single nucleotide polymorphism is the sequencing will be differ only at the point of one single nucleotide, and you have to also know about VNTR, variable number tandem repeats. So let's talk about this. We already know from Human Genome Project, thus 99.9% of human base sequence among the humans are same. 99.9% is same. Assuming the human genome was 3.3 into 10 to the power of 9 base pairs, how many base sequence would be difference? So it is the difference in the sequence of DNA which make every individual unique in their phenotypic appearance. So 0.1 difference will be there between each and every individual. If one aims to find out the genetic difference between two individuals or among individuals of population, sequencing of DNA is every time is daunting and very more expensive task, right? Imagine trying to compare two sets. You know one normal set, if you can compare between two. sets of 3 into 10 to the power of 6 base pairs. You can compare between the two sets. DNA fingerprinting is very quick way to compare the DNA sequence of any two individuals. Okay. DNA fingerprinting involves in identifying the differences in some specific regions in DNA sequence. We call that as repetitive DNA. So we already have seen that satellite DNA, repetitive DNA, variable number tandem repeats all are same, because in these sequence a small stretch of DNA is repeated many times. These repetitive DNA are separated from the bulk genomic DNA as different peaks during the density gradient centrifugation. The bulk DNA forms a major peak and other small peaks are referred to as satellite DNA. Major peaks and other small peaks, we call that as satellite DNA. So depending on the base composition like adenine thymine rich or guanine cytosine rich, length of the segment, number of repetitive units, the satellite DNA is classified into many categories. So such as microsatellites and mini satellites, etc. So the sequences normally do not code for any proteins. These will not code for any proteins, but they have a larger portion in human genome. The sequence shows high degree of polymorphism and forms basis of DNA fingerprinting. Since DNA from every tissue such as blood, hair follicle, they can take it from skin, bone, saliva, sperm, etc. So from an individual shows the same degree of polymorphism. So they become very useful in identification the tool in forensic application. Further, as the polymorphism are inheritable from parents to children, yes, that polymorphism is inheritable. So DNA fingerprinting is the basis of paternity testing in case of disputes and even in the crime scene also. As polymorphism in DNA sequence is the basis for this, polymorphism is only for the basis of genetic mapping in human genome as well as DNA fingerprinting. For genetic mapping and DNA fingerprinting, this polymorphism is only very essential. It is essential as we understand the DNA polymorphism in simple terms. Variation at genetic level arises due to mutation. So we recall the different kinds of mutations and their effects you have already seen in chapter 5, Principles of Inheritance and Variation. So in preceding sections of this chapter, new mutations may arrive in individual either in somatic cells or in germ cells that can generate gametes in sexually reproducing organism. If germ cells mutation does not seriously impair the individual ability to have offsprings who can transmit the mutation, they can spread to other members of the population through sexual reproduction. So we have the allelic, again recall the definition of alleles, the various molecular forms of gene. So sequence variation has been traditionally described as DNA polymorphism. If more than one variant at the locus occurs in the human population with a frequency of greater than 0.01, so in simple terms, if an inheritable mutation observed in population at high frequency, it is referred to as DNA polymorphism. The probability of such variation to be observed in non-coding DNA sequence would be higher, as mutation of these sequence may not have an immediate effect or impact on an individual reproductive ability. So these mutations keep on accumulating the generations after generations and form one of the bases of variability and polymorphism. So these variety of different types of polymorphisms ranging from single nucleotide change to larger scale. For evolution and speciation, such polymorphism play an important role, and you will study in detail in higher classes. The technique of DNA fingerprinting was initially developed by Alec Jeffries. He used satellite DNAs as probes, and that shows high degree of polymorphism. So we call that as variable number tandem repeats. It was called as variable number tandem repeats. So the technique is used earlier involved the southern blotting hybridization using radio labeled VNTR probes. It is included. So isolation of DNA. First we have to isolate the DNA, and after isolating the DNA we have to cut the DNA with restriction endonucleases or molecular scissors. So separation of DNA fragments by gel electrophoresis. So transferring the DNA fragments to synthetic membrane, which is nylon sheet or anything, and such as nitrocellulose or nylon. And hybridization using the labeled VNT, variable number tandem repeats probe. So detecting the hybridized DNA fragments by autoradiography, a scheme of representing the DNA fingerprinting as given. So the variable number tandem is belonging to the class of satellite DNAs, are referred to as mini satellites. A small DNA sequence is arranged tandemly in many copies. So the copy number varies from chromosome to chromosome. In an individual, the number of repeats show very high degree of polymorphism. So as the result of the variable number tandem repeats varies in size from 0.1 to 20 kb. Consequently, after hybridization with VNTR probes, the autoradiogram may have many bands of different sizes. These bands gives a characteristic pattern of an individual DNA. So they differ from individual to individual in population except in case of monozygotic twins. The sensitivity of the technique has been increased by use of PCR, polymerase chain reaction. So consequently, DNA from a single cell is enough to perform the DNA fingerprinting analysis. In addition to the application in forensic science, it is much wider application, such as determining the population, genetic diversities. Currently, many different probes are used to generate DNA fingerprints. So you can see here, this is chromosomal 7, chromosome 2, chromosome 16. So one is paternal and one is maternal. So here if you see, this is what is detected in the crime scene. In autoradiography, we can find out the person is B. So whatever the DNA we have taken, so we have suspected the two persons. In that, B is the person. So he is the one. From this we can find out, because the sequence, if you see, the same size will be there here. This is matched. So he is the one. So this is about DNA fingerprinting and Human Genome Project.